Welcome back to MD-Sim 360. Today, we're diving into an exciting MD simulation. First, a quick look at this powerful platform. MD-Sim 360 is an all-in-one platform for MD simulations. It covers system preparation, solvation, ionization, energy minimization, NVT and PT, and production run. No need to worry about compute power or analysis. The platform takes care of everything. For this video, we explored an interesting case: protein folding. This simulation captures the intricate process of protein folding in a short time frame. We ran the simulation for 15 nanoseconds, which took about 12 to 15 hours to complete. With MD-Sim 360's real-time monitoring, we accessed live results and trajectory data throughout the process. While the simulation was running, we analyzed the results in real time. Now, let's dive deeper into the findings. First, let's understand protein folding. Here, we have our protein, identified by its unique ID within a system containing approximately 50,000 atoms. Let's take a look at the full system, which includes ions and water molecules. Now, let's temporarily hide them to focus on the protein. Protein folding is all about the protein finding its most stable structure. It starts in an unfolded state, and as the simulation progresses, you'll see it slowly adjusting, reshaping, and settling into a more stable conformation, influenced by various factors along the way. By the end of the simulation, the protein moves toward a more stable conformation, though not its most stable state. Since we ran this for only 15 nanoseconds, it isn't enough for the protein to fully fold. Typically, folding takes significantly longer, sometimes over 50 nanoseconds or more. Next, let's get into the analysis. We'll start with the radius of gyration. Looking at the plot, you'll notice that in the beginning, the values fluctuate significantly, showing a lot of variation. This is expected since the protein is still adjusting. Over time, though, the values start to stabilize, indicating that the protein is settling into a more compact structure. So, what exactly is radius of gyration? Simply put, it measures how compact the protein is. It's calculated around the protein's center of mass, helping us track how folding is progressing. On the x-axis, we have time, and on the y-axis, we have radius of gyration. The blue curve represents the overall radius of gyration, while other components show variations in different dimensions. When the protein is more compact, the radius decreases, and when it unfolds, the radius increases. This is a key indicator of the protein's folding state. Researchers rely on this data to assess stability. If the radius of gyration keeps increasing, it could mean the protein is unfolding, which might not be desirable. One of the major advantages of MD-Sim 360 is real-time monitoring. Let's say you're running a long simulation, 200 or even 500 nanoseconds, and you notice the radius of gyration increasing rapidly. That could be an early warning sign. With live analysis, you can pause or stop the simulation, saving valuable computing resources and time. This makes MD-Sim 360 incredibly efficient, allowing researchers to make quick, informed decisions. As we look at the current data, we can see a sudden drop in the radius of gyration, which clearly signals that the protein is folding and becoming more compact. What's great is that MD-Sim 360 allows us to compare this in real time with the actual trajectory file. Yes, the trajectory is available live, which adds an extra layer of insight. Here, you can see the x, y, and z components of the radius of gyration. Around 20,520 picoseconds, the radius of gyration becomes more isotropic. The individual x, y, and z components keep changing, but overall, the radius of gyration remains constant. This level of real-time analysis is a game changer for understanding molecular dynamics. Next, we have the root mean square fluctuation data. There was a minor issue with the plot, so it's not visible, but the data is still available for analysis. Next, let's look at the hydrogen bond analysis. After 20,000 picoseconds, we don't see any major changes, just some localized patterns here and there. What we do notice is that when intra-protein hydrogen bonds decrease, the protein forms hydrogen bonds with neighboring solvent molecules. As we discussed in earlier videos, intra-hydrogen bonds provide stability within the protein, while protein-solvent hydrogen bonds are formed with surrounding solvent atoms. Between 26,000 and 28,000 picoseconds, we see a clear drop in intra-hydrogen bonds. But right after that, there's a sharp increase in protein-solvent hydrogen bonds. This tells us that some hydrophilic regions of the protein are being exposed, breaking their internal bonds, and instead forming new ones with the solvent. Now, let's check out the next graph. This is the salt bridge analysis. Looking at the trend, we can see that it remains almost constant throughout. There aren't any major fluctuations or significant changes in the data. Now, let's take a look at the solvent accessible surface area. This is a really interesting plot. We see a sudden dip right at the same point where we observed the change in the radius of gyration. This decrease is quite significant, meaning that residues that were previously exposed to the solvent are now getting buried within the protein. Essentially, this tells us that the protein is folding, causing some exposed residues to become hidden within the structure. Proteins have both hydrophilic and hydrophobic residues. Here, some of the residues that were in direct contact with the solvent are now being tucked inside as the protein becomes more compact. The sharp drop suggests that a major structural shift, possibly a fold, occurred at this moment. This is a crucial metric for understanding how the protein structure evolves over time. Now, let's correlate these two graphs, the solvent accessible surface area and the radius of gyration. Before the folding event, both graphs show fluctuations, some increase, some decrease, but after that, they stabilize. The sharp drop in both values at the same time is a strong indicator that folding has occurred. When we see proportional changes across multiple analyses, it reinforces the conclusion that a significant structural shift is happening. Of course, we'll confirm this by analyzing the actual trajectory, but just from the data, it's clear some kind of folding is taking place. The key folding event happens at 20,000 picoseconds. Here, we see a sudden drop in the radius of gyration and solvent accessible surface area. This suggests the protein overcame an energy barrier, allowing it to collapse into a more stable conformation. This isn't a gradual process, it's an immediate shift, meaning the protein didn't just slowly fold. But instead, once it overcame that barrier, it rapidly transitioned into a stable state. This is a crucial phenomenon in molecular dynamics. Protein folding plays a key role in ligand binding, docking, and even diseases. In many disorders, protein misfolding or unfolding can be a major issue. Some proteins in the body lose their structure, leading to dysfunction. Understanding this process is critical in biomedical research. What's fascinating is that small proteins can fold within microseconds, something we can't fully simulate due to computational limits. But even in this time frame, we can see how quickly structural changes occur once the right conditions are met. And it's not just the protein that's involved in this process, the solvent plays a crucial role too. In this case, the solvent is water, and it actively participates in protein folding. If you look closely, you'll see a lot of atoms surrounding the protein. These are water molecules. Water isn't just surrounding the protein, it actively shapes folding by stabilizing or disrupting interactions. Now, looking at the other graphs, most of them remain unchanged. But if we check the Lennard-Jones short-range interaction, we do see some interesting changes. At first, The interaction decreases for a bit, but after that, it stabilizes. This tells us that short-range interactions within the protein have settled after it reached a stable conformation. Now, using the trajectory, we can actually see how the protein folded, which is crucial. Because when proteins misfold, it can lead to serious diseases like Alzheimer's and Parkinson's. These simulations help researchers understand why misfolding happens, what factors contribute to it, and how energy and environmental conditions influence the process. Now, let's take a look at the trajectory. This may take some time to load as the data is being converted to MMCIF format. Once the trajectory file is loaded, we can see we have about 2,000 frames. Here's how the calculation works. For every nanosecond, MDSIM 360 generates around 50 frames. Since we've run the simulation for approximately 40 to 50 nanoseconds, this gives us about 45 to 50 frames. So, while the exact number can vary, the maximum frame count is typically around 50. Let's play the trajectory now and observe how the protein behaves over time. At the beginning, you can clearly see the protein adjusting, trying to find the most stable state. During this phase, there aren't any significant changes happening in the protein structure. It's mostly in a transitional phase. But as we continue, we notice a significant change. The protein undergoes a major transformation and begins to fold. From here onwards, you can see that the structure doesn't change much. Once the protein has folded, it reaches a point where it becomes very stable. This is the final conformation, and it definitely looks much more stable than the initial one. I believe this compact structure has led to a decrease in the solvent-accessible surface area. Now, let's compare this with the original structure. This is the final conformation of the protein. It is much more compact and stable. Now, comparing it to the original structure, we can see that the protein was initially much longer before folding into this condensed state. This compact structure indicates that the protein has likely reached its lowest energy state, making it more stable. This observation perfectly aligns with what we saw in the graph, confirming that the changes in solvent-accessible surface area and structural stability are clearly reflected in the trajectory itself. This real-time visualization is one of the best features of MDSIM 360. Not only do we get detailed graphs, but we can also observe the trajectory in real time as the simulation progresses. Around the 20 nanosecond mark, I observed the protein fold into its stable conformation in real time, highlighting MDSIM 360 as a powerful tool for molecular dynamics analysis. That wraps up the video. Share your questions or thoughts in the comments.